Okay, let's have a class. And last lecture before the exam one, some of you ask a question how we prepare for the exam. Obviously, you're going to look at your slides. It will be slides based. My notes will be for you to understand it. And I also going to post on the eCampus the practice exam tonight. Then you have a weekend to do the practice exam, okay? The next week, Tuesday, we're going to talk about all those practice exams. And our real exam will be similar and also based on our practice exam. So that will be certainly a learning material for you to prepare for the exam one. Okay, let's talk about what we have. This is the key we have last section. Is bacteria growth curve. X-axis is the time, Y-axis is bacterial population, could be cells per ml, and the curve looks like this. So we have a lag phase, log phase, stationary phase, followed by the death phase. This is a bacteria growth curve. And the death process, we also call it a programmed death. Then we talk about the factors affected the bacterial growth. We mentioned about the first one is pH. Because of the pH, we could say a acidophile. We have a neutrophile. And we also have a alkaline file. This is a pH level 0 to 5.5, 5.5 to 8.0. It's more than 8.0. Then we have a temperature we mentioned about. Because temperature is highly related to their enzyme activity. We have cyclotropes, we have cyclophile, we have a mesophile, then followed by thermophile, and last one is hyperthermophile. No, say super thermophile, hyperthermophile. And corresponding to the temperature, this is 0 to 30 degrees, this is 20 to 45 degrees, this is 40 to, this is 55, this will be, the first one is 0 to 30, then 0 to 20, okay, 0 to 30 degrees Celsius, 20 to 45 degrees Celsius, and you have a 55 to 85 degrees Celsius, the last one is 85 to 113 degrees Celsius. Okay, that's a temperature. Then we mentioned about something important like acid. Okay, acid related to the pH, but more is acid resistant problem. Then we mentioned about is water activity related to moisture content. Okay, that's all we mentioned about the bacteria growth curve. Then we talk about halophiles, extreme halophiles. So we end up with the oxygen amount. Okay, so let's talk about, start from here. We said the oxygen amount, said very simple, has three categories. Aerobic, anaerobic, and facultative. Okay, what is aerobic? They must use oxygen to grow. Anaerobic, no oxygen. Otherwise, dead. Said very simple. One is facultative. Grow with or without oxygen, but better with oxygen. And grow better with oxygen. Aerobic bacteria like Pseudomonas, anaerobic bacteria like Clostridium. Facultative, majority of the foodborne pathogens are facultative. Okay, now we're going to talk about this. What is the impact of the oxygen for a bacteria? It surprised a lot of people. The impact of the oxygen for bacteria, somehow, it is a toxin. It is not really helpful too much. This is going to go back to talk about the electron suborbital arrangements. I do not know whether you learn in your biochemistry or your chemistry class, what is it going to be looks like? 1s2, 2s2, and 2p4. So what it looks like? 3 boxes. Little chemistry, you will be understanding up, down, up, down, up, 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 then down. Why its arranged looks like that is following Pauli exclusion policy and Hans rules. So these are the basic the knowledge of the chemistry of the electron orbital arrangement. The problem for oxygen is right here. Based on Pauli exclusion policy and Hans rules, they have, this is we call it an unpaired electron. All these are saturated, this is unsaturated. So what happened? Once you have unpaired electron, the electron could go, the electron could come in. So what happened? Oxygen could be working with this, become, this is a toxin. And this guy could also further become, with water, hydrogen peroxide. These two are toxic material. So bacteria survive during their living environments. They must have something to hydrolyze these toxins. What are they? This is SOD. Superoxidase dismutase. It's an enzyme. Could hydrolyze the superoxygen become oxygen. Why this is a toxin? You have these empty vessel could have an electron. This is originally in an energized stable status. Once you come electron, this become highly energized, which means they're going to do something. They usually do a bad stuff. So that's a toxin. Then the bacteria must have the enzyme to hydrolyze it. If they have a superoxidase dismutase, that's good. H2O2, catalyst. H2O2, you will learn that is hydrogen peroxide. That is even antimicrobials. So if you have a catalyst, become oxygen and water. Therefore, whether bacteria can survive with or without oxygen is related to whether they have these two enzymes, which is SOD and catalyst. So based on the amount or the existing of SOD and the catalyst, they categorize all these 3 major categories. Now, later on, people are going to do more research what they find. They find something called the aerotolerant. What is aerotolerant? This is grow equally well with or without oxygen. Remember, facultative can grow without oxygen, but grow better with oxygen. Allotolerant is grow equally well without oxygen. Now you had anaerobic. You cannot have oxygen. Oxygen is a toxic. But there is something can grow with limited amount of oxygen, but cannot be more. They're called microallophilic. This usually is 5 to 10% oxygen. If more than 10%, bacteria die. Microallophilic is a very tricky environment. It's related to an important pathogen in the poultry meat, which is Campylobacter. And it is very ironic. Campylobacter, because it is microallophilic, it is difficult to grow in the lab. If your cultivation, Campylobacter, just in tropic soil broth or nutrient broth like you did in the lab, it will not grow. However, it is a big risk in the poultry industry. So it's very ironic. It's because it's only need 5 to 10% oxygen can grow. And it's limited condition for that. So what happened? It's very interesting. On the poultry skin surfaces, we always get isolation of Campylobacter. But in the ground poultry meat, we never find it or barely find it. So it's very tricky condition. Now, how do we cultivate aerobic bacteria? Aerobic bacteria will just grow, but it will stay on the top of the tube. Anaerobic bacteria, we need tools. It could be a chamber, like what you showed on your slides. The chamber is expensive. Usually it's $20,000. We will be pumping nitrogen lots of the time to create sometimes a vacuum system without any oxygen. Now we also could be connected to the different gas supply. Put appropriate amount of the carbon dioxide and make it 5 to 10% of oxygen. Then create a microallophilic environments, and the Campylobacter can grow there. And we also sometimes in the lab, we don't have that $20,000 to buy. Let's say we don't have a $500. What we do, we're using a jar. So the jar is called anaerobic jar. How we do it? We put inside is a gas packer. The gas packer, we are adding 10 ml water. It will generate carbon dioxide and hydrogen. So how we decrease the amount of the oxygen? Hydrogen and oxygen will react to generate water. And we also want this reaction to be quickly. So on the edge, at the bottom of the lock screw, we have a catalyst chamber. We want this reaction happen quickly. Now finally, how do we know the oxygen is gone? We need to add some of the indicator. Most of the time we're using methylene blue. So methylene blue is a dye we mentioned about in the simple state. It is also an indication of oxygen. If no oxygen there, the methylene blue is colorless. We can also use another chemical called resazarine. If it's colorless, the resazarine is colorless, it is also means no oxygen. Okay, so these are the two different ways we can cultivate an anaerobic bacteria. My lab, we do this anaerobic jar. The jar is also expensive. It's like $500. It's just an empty jar. And this gas packer is like pretty much $5, $10 one pack. And how many plates we can put? About like 12 plates. So if we want to do a whole day for Campylobacter analysis, we maybe need to use 8 or 10 jars. Campylobacter is a microallophilic, it's the same thing. We're using a gas pack to create microallophilic environments instead of like this to take out all of the oxygen. Okay, so that's what's anaerobic. Biofilm, we already talked about the biofilm. So what happened is usually in the bench, like this, we didn't do a good job of the sanitizing or cleaning. Let's say I prepare ground beef. And on this bench, if I didn't do a good type of the sanitizing, more or less we'll have some moisture. And we already talked about it. As long as there's some of the moisture will be transferred, become water activity, then the bacteria will grow. Now how they grow? Of course they grow poorly. So they grow a little bit, and they grow a little bit. They connected with each other by van der Waals force. It's loosely attached. And some of them will be dislodged, which means released to the environment immediately. But the more and the more they're going to accumulate. Because cell and cell could do a communication called coring sensing. More and more cells can communicate or grabbed with each other, they will be filled a matrix, which is a polymer matrix composed by polysaccharides. Once the matrix has been formed, it's highly resistant to antimicrobials and sanitizing and detergent. So I talked about that before. Normally the bacteria, we call it vegetative cells, only need 50 parts per million chlorine. If it's biofilm, you use 500 parts per million chlorine, may not kill them. Okay, and also the biofilm could be on the bench forever. It could be stay there for a week, a month, or a couple of years. The only thing what you can do is intensively scrub and followed by rinse using antimicrobials and rinse again. There are multi-steps. We call it a multiple hurdle treatment to finally kill the bacteria. It is very difficult to do. That's why the biofilm deserves to do the research. Can use scanning electron microscope to see the 3D images or using laser confocal microscope. That's what we talked about. Because it is difficult to remove, we need to keep our good sanitation standard operation procedures. That is called SSOP. Okay? So just want to give you some introduction for the biofilm. The major part of this class, we want to talk about the bacterial media. In the exam, we will have some questions for that. And also, next week, Tuesday, we will be to do some of the lab work with selective media. Lots of the topic we can talk. First thing first, growing bacteria, we need some material. We call it a bacterial media. Therefore, grow, transport, storage microorganisms. The difficult part is the category. So, based on chemical characteristics, we could differentiate all the media into defined media and the complex media. Now, what is defined media? Every single chemical ingredients are well described, including the name and the amount. Okay, the key thing here is every single chemical. So this gives you a question. Okay, now what is complex media? Otherwise, it is complex media. Okay, that's simple. Okay, what is the misdefined media? I give you a media. I have water, 1 liter, sodium chloride, 2 grams, peptone, 10 grams, traptone, 5 grams, beef extract, let's say, 1 gram, let's say. If this is a media ingredients, is this a defined media and a complex media? How many of you think this is a defined media? None of you? How many of you think this is a complex media? Oh yes, this is a complex media, good. Why? Remember, the definition for the defined media is every single chemical ingredient. It looks like it's described, but are these every single chemical? No. We don't know what inside of the peptide. Peptide is a protein extractor. Tryptophan is also a protein extractor. We don't know what's a chemical inside here. So, this is an example of a complex media. But what is an example of a defined media, or we say synthetic media? Look at the top. Every single chemical needs to be well defined. Can you see it right here? You have every single chemical well defined, their name and amount. This is BG11 media is very good for cyanobacteria, which means the bacteria could be using light as an energy resource. This is also a very basic media to grow biofilm in the lab. Biofilm usually need to grow at the synthetic or defined media because in the poor nutrition status it's easy to fuse the biofilm. So you grow in the synthetic media first, then you spread onto the coupons, like stainless steel coupons. It's very easy to fuse the biofilm. Okay? Look at here. Neutron blast, what we did in the lab. Peptide, gelatin hydrolysate, beef extract. That's a complex media because we don't know exactly what's inside the peptide and the beef extract. Tryptic soil blast. We don't know what is inside of the peptide and the tryptophan. Macanchi agar. Well, looks good, but we don't know what is called a digestive casing. What is detailed inside of the digestive animal tissue? Because it could be millions of different chemicals there. The chemicals is not well defined. Okay, so that's defined media and a complex media. The second category, relatively easy, is based on physics characteristics. We'll have a liquid media, we have a solid media, and we have a semi-solid media. Okay, liquid media you already see in your lab, which is broth or neutron broth. Solid agar you already see in your lab. It is agar or slants. Agar plates or slants. Semi-solid we will do one time in the lab. Contains 0.5% of agar. It used to detect bacterial motility. If bacteria no motility, they stay in the middle. If there is a strong motility, which means it's petrotrichous, it's going to go everywhere. Okay? If it stay in the middle, which means monotrichous. There is only one tail at one end of a flagella. So semi-solid agar will do once in the lab. So most of our lab material is liquid media, semi-solid and solid media. The most difficult part to understanding is based on the function. First category, support media. Okay, what is support media? Support most which threaten bacterial cell to grow. You can say lots of the time, 35 degrees Celsius, 24 to 48 hours, they could grow approximately 10 to the power of 8 cells per ml. A good example, tropic soil broth. What you did in nutrient broth is a good example. Tropic soil broth, it is also a typical supporting medium. What means vegetative cells are just general common bacteria like E. coli. Okay, it's a supporting medium. Second, enrichment medium. What means enrichment medium? Support bacteria to grow dramatically. How dramatic it is could be around 10 to the 9 to 10, the power of 10 cells per ml. What is a good example of enrichment medium? Blood agar. Blood agar is completely looks like red. That picture now very good. Looks like it's purple. We'll talk about that. Blood agar. Blood agar. It is basically tropic soil agar base with 5% sheep blood. Now, why they do not use human blood? Because back in 1970s, the scientists did the research. They find that the 5% sheep blood has the same characteristics with human blood. So in order to save the cost, and it is also ethically easy to get, the blood agar is always tropical soil blood, adding 5% sheep blood. It is very rare. You will see next week Tuesday. Looks like your blood. Bacteria on the blood agar can grow dramatically. Huge. That's called enrichment media. Now why we want to use it? In the clinical area, we want to get a sample from the patient. And we want to know what type of the pathogen it is. Obviously, we want it to grow dramatically. We don't want it to grow only 2 cells, 1 cell. We cannot see it even. We want it to grow as many as we can. We can do the identification and followed by some of the analysis, like a biochemistry analysis. Okay? So that's a blood agar. Enrichment media, I'm going to also mention another agar. It's called chocolate agar. This name is very, very nice, but very confusing. Chocolate agar. Chocolate agar, there is no chocolate there. It is not you make an agar that you put a chocolate candy ball and then become a chocolate agar. It is, should say, chocolate like agar. So how do you do it? They make TSP base, they add 5% sheep blood, then they heat it at 70 degrees Celsius. After doing that, it turns brownish color. Looks like a chocolate. You look at that, that's like a chocolate. That's why it's called a chocolate agar. Now, people think you guys are smart. They took this out. That's become chocolate agar. But the full name should say chocolate-like agar. Chocolate agar is also an enriched media, especially for fastidious bacteria. What means fastidious bacteria? Very difficult to grow. in the clinical area, there are some of the bacteria. It is cause pathogenic impacts, cause disease, but it's very difficult to grow. Haemophilus influenza, Neisseria gonorrhea. Like that. That's why we need a chocolate agar. I have one sample. I will show you on Tuesday for the chocolate agar. Chocolate agar is expensive. TSA, maybe $2 if you buy. Blood agar maybe $3 to $5. Chocolate agar, one agar, $7 to $10. It's an expensive one. Okay, it's an enrichment media for fastidious bacteria. So that's a second category. We talk about support media. We talk about enriched media. Number 2. Number 3, selective media. What means selective media? The media containing certain ingredients favor certain bacteria to grow. Others inhibit. What is an example? I'll give you a very simple example. Is tropic soil agar. Add 100 parts per million penicillin. This is a selective media. What type of the bacteria will grow? Only penicillin resistant bacteria can grow. That's a selective media. That's a very typical example. Okay, that's called a selective media, favorable certain bacteria to grow. Last one, differentiate media. What is differentiated media? Bacteria grown the agar will have colony color change based on biochemistry reaction. A very good example is blood agar. It could be alpha hemolytic, a beta hemolytic, and gamma hemolytic. Alpha hemolytic, dark green color. It is incomplete lysis blood cell. Beta hemolytic, transparent bacterial colony because it is complete lysis blood cell. Gamma hemolytic, nothing happened. Okay, we mentioned about all these different types of the bacteria, media. What is the most difficult part? Look at the blood agar. Blood agar is an enrichment media. It is also a differentiated media. What this means? Which means the bacteria we are using in the lab, it is not fit one category. It fits multiple different categories. That makes it a little bit difficult. So, we're going to give you some examples. This will be an example, a very good one, Macanthi agar. we will do in the lab, but today we'll give you some examples. Okay, Macanchi agar. When we talk about the agar plates, we look at the ingredients. So here are the ingredients. So peptide is just for bacteria to give some nutrients. Salt keeps the osmotic balance. We have agar. We'll have to form the solid medium. We need agar. Okay? We need a neutral red, which is a pH indicator. We're going to talk about that. Macanchi agar. It is a selective medium. Why? Because it contains a very important chemical you already know. Crystal violet. Why it is a selective reagent? Remember your gram stain? Crystal violet will firmly attach to peptidoglycan. It showed a purple color. What this means? Of course the bacteria is died because it's already penetrated to go to the peptidoglycan. So, Macanchi agar, because it contains crystal violet, so only gram-negative bacteria can survive. Understandable, because crystal violet attacking peptidoglycan of a gram-positive cell. This is a very good selective media. Using crystal violet, you at least taking out half. You have left 50%. Okay? So that's a selective media for gram-negative bacteria. Second, Macanchi agar. If we just say Macanchi agar, it has a very important component. It's called lactose. Now, we will learn later on. Lactose will go through fermentation. You understandable, some of you cannot drink milk. You will have a diarrhea, abdomen pain, or all kinds of things because of the lactose fermented intolerant. Is that right? So lactose will go through fermentation. Lactose fermentation. On the agar plate, it's going to be positive or negative. If it's positive, it is pink. If it's negative, colorless. Why? Lactose fermentation will generate acid. And I have a pH indicator called neutral red. And the neutral red at a lower pH, it is pink color. So the lactose fermentation. Let's say E. coli. It is lactose fermentation positive. So it is pink on Macanchi agar. Macanchi agar, it should be called Macanchi lactose Macanchi agar. Why? Because lactose could easily to replace with another carbohydrate called sorbitol. Why sorbitol it is important? If sorbitol Macanchi agar it can use to differentiate E. coli 0157H7 versus non-0157 Shigella toxin generated E. coli. And I tell you the results is very interesting. What it looks like. Lactose macanthi, this is. This is sorbitol macanthi. What's the results looks like? Let's say this is E. coli, 0157H7. This is STEC, non-0157. This is pink. This is colorless. For non-0157, this is colorless, and this is pink. Sticky ends. It's very easy to differentiate non-0157 Shigella toxin general E. coli and 0157H7. If you look at the news, you will know these are the major two pathogens in the United States for beef products. You hear about those all the time. We have 500 pounds, 5,000 tons of beef in Indiana. A plant is contaminated, recalled, or outbreaks with E. coli 0157H7. We have another 1,000 pounds in some plants recalled because of the potential contamination with non-0157 Shigella toxin E. coli. Using macanthi agar very quickly can do a determination. So, basically we say a general macanthi agar, we should say lactose macanthi agar. Again, people think you're smart, so take it out. But not really. And so what they do, if it's a general macanthi agar, they call it a macanthi agar. The carbohydrates is lactose. Once they contain sorbitol, they will say sorbitol macanthi. Okay, so they do like that. But for you to understand, a general macanthi agar is a lactose macanthi agar. Same as chocolate agar. There's no chocolate. It's chocolate-like agar, but they take the like out. It looks like a branch. Okay, so that's a macanthi agar. What it looks like, that's what the pink color looks like. And you will see a little bit of transparent zone there. This happens for E. coli on the macanthi agar all the time. E. coli has a light beta hemolytic function. So it looks like a surrounded transparent zone. Okay? We already talked about differentiated media. This is important. Okay. Sorry, we just want to make sure one more time. Macanthi agar, selective media, differentiated media, lactose fermentation. Positive pink, negative colors. In U exam, we will have some tricky question for the selective media and differentiated media, but we're also going to practice over the weekend when I send out the practice exam question tonight. Next week, Tuesday, in the lab, we're also going to introduce you another media, which is mannitol salt agar. But we'll have a question in the practice exam first. So you get to know another very typical selective media and differentiated media called mannitol salt agar. Why it is important? Because it used to determine Staphylococcus aureus. This one is for E. coli, another one is for Staphylococcus aureus. Okay, we'll introduce you in the practice exam questions and also in our lab next week, Tuesday. This is a very important slide. This tells you blood agar is a differentiated medium. This gives you two good examples. The top one is beta hemolytic. The clear, transparent zone. A typical example, Streptococcus pyogenes. This is the one called strep throat. I already have the email from some of you said I'm not going to come into the lab because I have a strep throat. And I expected that every semester. Next one. You see the dark green color there. It is alpha hemolytic, incomplete hydrolysis, hemolysis of the blood cell. Good example, Streptococcus pneumonia. This is the one. I showed you the picture before. Having capsule stain. It's a heavy capsule. It is number one ammonia caused in the community. We call it the number one community acquired pneumonia. So Streptococcus pneumonia, alpha hemolytic. These slides we will talk again on the exam too, because it's a very typical one. Show you two major pathogens using blood agar can do a very quick, simple clinical identification. Okay. Isolation of pure culture. Bob Koch's group created a media using an agar as a solid media to isolate a pure culture. So, we already been practicing the lab, streak plating. You already did it. The first lab, you already see your results. I already show you my results. I have 3 plates really good, 1 is not good. Lots of you have good results. You will see that. You will see a single, very clear colony represent a pure culture after streak plating. Okay, so that's what this one I skip it because you already did it. Your plates, if it's good, very similar to that one. Because you have a Serratia, remember? Serratia is pink to red at 25 degrees Celsius. And then E. coli should be colorless or white. Lots of you, the place looks like that. It's a really good job. Spread plating. We will practice this in the lab, so I'll do it relatively briefly. Spread plating is we're going to preparing lots of the tube. When we prepare lots of the tube for serial dilution, and finally, it looks like this. It could use 9.0 ml or 9.9 ml, which means 10 or 100-fold serial dilution. After that, each tube needs to do vortex, but we don't have it, we just do shake. 15 seconds. Then we use a spreader to spread on the agar surface. This is what you're going to find. You will find on the agar plates, you have all these things are well designated. This is called CFU, stands for colony forming unit. But I want to emphasize, this is not one cell. This is one colony. How many cells there? More than 10 to the power of 6. Okay, that's a colony forming unit. Especially this will represent a pure culture. So how are you going to do a calculation? Just count. Like you count an apple, how many it is. But you will say, on the agar plates, there are so many dots. How are you going to count it if it's so many? So in the clinical area or in the microbiology area, we set up a standards. We want you to do, the colony is within 30 to 300. More than 300, we usually don't count it, because it's too many. We just write TNTC, too many to count, because it's one accuracy. Less than 30, we also don't do it, because it's may coming from contamination. In order to reach 30 to 300, what we have to do is serial dilution. That is what we have on the picture here to do serial dilution. You could do tenfold, use 9.0 ml buffered peptide water, which is 0.1% buffered peptide water, or you use 9.9 ml. That means it's 100 serial dilution. Okay? These two, we will practice in the lab. That's a spread plating. Okay, what is pour plating? Spread plating is you prepare the agar. Then you add a bacterial liquid solution, you do the spread. Then you do the incubation, you see the colony. What is pour plating? You add the bacterial solution on an empty petri dish. You pour melted agar and you mix it. So the colony will form usually later on. Lots of the time at the bottom, on the surface, or even inserted in the middle of the agar. That's called pour plating. But why we have to do pour plating? Because pour plating saves the time. Spread plating, we need spending a whole day to make the bacterial agar medium first. pore plating, we can make the media and do the bacterial enumeration at the same time. But the pore plating has a disadvantage. Because of pore plating, we are using melted agar. The melted agar usually has 55 degrees Celsius or 50 degrees Celsius temperature. So the mild heat could kill bacteria. Okay, so usually you get the results of pore plating is much lower than the spread plating. So pore plating is only for the food industry. They do a quick identification. How many bacteria are there? Spread plating and pore plating we will practice after next week. We have Tuesday to do spread plating, Thursday to do pore plating. We want to use E. coli as an example. Okay? Spiral plating, this is something just to introduce. It's expensive. This equipment, right on the top, somehow is $20,000 to $50,000, $20K to $50K, more than your tuition fee in WVU. What is it used for? You have those three vessels. The first one is alcohol. Second one is water. These two for cleaning the pipes. So this is the sample. This is the vacuum system. When you click it in, it generates a vacuum system. After wash, they abstracted the sample into this thin tube. And then they do a stir. Then they have these type of thing generated. This is use crystal violet as a demonstration. Then you do the incubation, you see the colony. And then we're not going to count it. We put into an automatic machine. It connected to the computer, it will show you results in 5 seconds. Now why we do spread plating? Why we do spiral plating? Spiral plating saves the dilution tubes. Spread plating, we need to use so many tubes. Look at this, how many tubes we need? 5 or 6. Spiral plating, we only need to use 1. It covers 2 to 4 dilutions. But there is a limitation. Spiral plating can only test in liquid, which is a pure liquid products. Not even a juice, a popped juice, because the food particle will stuck there. So there's a limitation for that. In the old time, this is in the old time, the spiral plating, what they do. They don't have a machine, they have these type of the chart to cover it, then you count it. Just want to let you know in the old time. And this is really good. This is a typical results of the spiral plating, what it looks like. This is the one you will do in your lab. Somebody already did. Remember, we have E. coli, we had Celosia, and I ask you to do the work is compare the colony morphology. And I want to emphasize once again, this is talk about is colony on the agar plates. That's called a colony morphology. You gram stem results. That is called cell morphology. It's a bacterial cell after gram stem. It is different. One is on glass slides, this is on the agar plates. And you will do this in your lab for the unknown number 1. So I just skip that part. Last section, measurements of microorganism counts. We have several methods can do. This is very similar to what you did for your blood cell in your biology lab. You put in a chamber, you put a bacterial solution, then you count it. But what's the problem for this? You cannot differentiate live and dead cell. This you can. When you see the colony there, it's a live cell. When you use this method, you cannot know it because it's all messed up. Okay? You also can test the turbidity. Remember, in the lab, I will say everybody look at your tube. We have 3 markers for bacterial growth. Number 1 is turbidity. But how turbidity it is, we can use a color spectrometer to do the measurements. If you see lots of the bacteria, the absorbance value usually is higher. If it's Not many bacteria, but start to grow, the absorbance value is lower. But again, this is only give you a brief idea. You cannot differentiate live and dead cell. Last one did this, what I did in the lab a long time ago. So what happened is, the company called Fisher Scientific, they generate a tool called isograde. The membrane filtration system. This used to testing Legionella or some of the fungi in drinking water, which is municipal water. Because based on the EPA regulation, our drinking water is zero tolerance for coliform and the fecal coliform. So we do the test. When we do the test, we use a funnel. And we put a membrane filter there. The membrane filter is very tricky. 0.45 micrometer. This is the size will let the bacteria penetrate, will let the fungi penetrate, and bacteria actually could prevent it. If the size decreased to 0.22 micrometer, then most of the microorganisms could be prevented. So filtration is one of the methods to do the disinfection or sterilization of the bacteria. So once you go through the membrane filter, then the membrane filter take it out, and you put on the agar plates, and you do the incubation for 24 hours at 35 degrees Celsius, you will see the colony there. If you see your drinking water sample looks like that, it's terrible. That means it's molds and yeast everywhere. When you see these type of things, that's molds. When you see here, it looks like yeast. This is certainly any drinkable water is failed. This is huge. And you can just go ahead and count the colony how many it is. It's like 50 or 60. If the dilution factor is 10 to the minus 3, that means you have 50,000 cells there. It's huge. Certainly, the water system is contaminated with animal poop. That's very typical. Animal fecal sample, said very scientifically. Okay, so that's something interesting. And this system, like $2,000, $5,000 ish. This is a vacuum pump. This is the liquid for the solution, and you go in slow, and you get the results. Then finally, the colony usually see very good. It's better than anybody else they do. They on this film, they averaged very well, and it's very easy to count. That's called the isograde, the membrane filtration system for testing fungi on drinking water. Okay, so that's what we have for the exam 1. 4 slides, 4 whole lecture slides, and we'll post the practice exam tonight.